You can look at the structure of silica, which is presented here, and this is having active OH groups. These groups are called as selenal groups, and these selenal groups are really responsible for the adsorption of the solutes over the surface of the stationary phase. And if these OH groups are many over the surface of the stationary phase, there are more number of hydroxy groups are present over the surface of the stationary phase. We cannot expect a proper interaction of the solute with the stationary phase because these OH groups may become responsible for the physical adsorption, and this physical adsorption may lead to increased band broadening. So in order to eliminate this problem, these OH groups must be inactivated or deactivated, or they have to be blocked. This will be done by chemical treatment. As I have already explained under gas chromatographic technique, the chemical treatment will be done by treating the stationary phase with the reagents such as dichlorodimethylsilane or hexamethylsilazane in order to get disillyl ether. This is a disillyl ether. And this chemical treatment will also be done followed by washing the stationary phase with an acid or alkali in order to remove the trace metal impurities. Otherwise, the trace metal impurities are going to interfere. They are going to impair the resolution. They are going to impair the separation. That's what it is very, very important. So the nature of the stationary phase, I mean to say that the polarity of the stationary phase is decided by the functional groups which are attached to silicon of silica. You take this silica, this is a disillyl ether of modified silica. It is having R group here, and this R may also be contained with R group, and this R group can be regulated in order to vary the polarity of the stationary phase. If you assume, if you assume the modified silica by using methyl substituent, octyl substituent, and octadecyl substituent, this stationary phase has been used for the separation of, say, 6 compounds which are presented here, namely uracil, phenol, acetophenone, nitrobenzene, methyl benzoate, and toluene. If you subject these 6 compounds or mixture of these 6 compounds for the separation by HPLC by using 3 different stationary phases such as methyl silica, octyl silane, or octadecyl silane. So this methyl silane or octyl silane or octadecyl silane are derived from the stationary phase what I have presented here. In place of this R, if you put methyl group, this is going to be methyl silane. In place of this R, if you put octyl group, this is going to be octyl silane. And similarly, if you put C18H37 here, this is going to be octadecyl silane. Octadecyl silane. So 3 different stationary phases were used in order to separate these 6 compounds separately. While using methyl silane, 8 compounds were separated, but the degree of resolution or the rate of separation of 6 compounds was not satisfactory. Even the separation is over within 8 minutes. You can look at the chromatogram. Although 6 compounds have been separated successfully, the separation is over in 8 minutes, but there is overlapping. There is more overlapping between the peaks of individual solutes, neighboring solutes. So as a result of this more overlapping, we cannot expect the resolution, and therefore the separation process is not going to be successful. If you change the methyl group of the silica by octyl group, if you change methyl silane by octyl silane while using the stationary phase for the separation process, by using octyl silane as a stationary phase, you are able to complete the separation within 16 minutes because 6th compound has been eluted in 15 minutes. So 16 minutes will be the maximum time. The overlapping which was seen in the separation of these 6 compounds while using methyl silane as the stationary phase was diminished while using this octyl silane as a stationary phase. If you look at here, the chromatogram, there is only less overlapping between the peaks of solute 3 and solute 4. But there is no any overlapping for the peaks 1, 2, 5, and 6. The resolution is somewhat improved here while using octyl silane. If you replace octyl silane stationary phase by octadecyl 1, further we have increased the resolution to a maximum extent. The resolution is highly excellent, highly satisfactory, acceptable. but the analytical time was extended to 24 minutes. So, however, irrespective of the analytical time, if the resolution is okay, if the resolution is acceptable, that chromatographic procedure is going to be adaptable in any laboratories, because that analytical procedure is going to produce achievable separation within the stipulated interval of time. Therefore, for the separation of these six compounds, octadecylsilane is going to be the most suitable stationary phase over a methyl or octylsilane stationary phases. And this can be explained by looking at the chromatograms obtained. There is not even 1% of the overlapping seen between the two neighboring peaks. If you look at the peaks 1, 2, 2, 3, 3, 4, 4, 5, 5, 6, etc. If you look at the neighboring peaks, there is no overlapping at all. As a result of this, we can conclude that the octadecylsilane is going to be the most suitable stationary phase for the separation of these six compounds. So, and this is going to give you the key that when we discussed about the optimization of the chromatographic condition in order to improve the resolution, we have discussed where in liquid chromatography and in gas chromatography, the type of the stationary phase may be changed in order to increase the resolution. So you can change the octylsilane to octadecylsilane. Before octylsilane, you have used methylsilane, so you have failed to obtain the required resolution. Therefore, you have moved to octadecylsilane, you have achieved the required resolution. Therefore, the separation has become efficient for the separation of these six compounds. So, here also I have presented many chromatograms for the separation of three compounds which are present in a mixture by using acetonitrile water 41 is to 59 mixture of acetonitrile and water if you use, which is having a capacity factor of 5. But there is more overlapping between the peaks of six compounds 1, 2, 3, 4, 5, 6, and these six compounds which were shown as given in the previous slide. So overlapping is available here. And if you change the composition of the mobile phase in this respect, 30% acetonitrile and 70% water, the capacity factor has been increased to 10, but overlapping has not been removed. Similarly, you look at these chromatograms also. So you can have a clear picture in this last chromatogram that by using the mixture of acetonitrile, tetrahydrofuran and water, the capacity factor can be obtained to value of 10, but all those six compounds have attained the resolution with lesser overlapping. If you look at here, overlapping is more. In the fourth chromatogram, the overlapping is more because only mobile phase of THF and water was used. But here, the mixture of acetonitrile, tetrahydrofuran and water is used as a mobile phase, and by means of using this particular mobile phase, we have succeeded the separation in order to separate all those six compounds. So this particular slide and the previous slide indicating that the separation efficiency in HPLC or liquid chromatography can be enhanced by changing number one, the stationary phase. A suitable stationary phase must be chosen in order to succeed the separation. Then we can play with the mobile phase. We keep on changing the composition of the liquids which are used to prepare the mobile phase, and even we can regulate the pH of the mobile phase. And even if we go one step forward, temperature can also be regulated in order to increase the resolution. So this you should keep in your mind. And after having the knowledge on isocratic illusion and gradient illusion in liquid chromatography, gradient illusion will be performed for the illusion of the solute molecules by using a single solvent or a mixture of a constant composition of two different liquids as a mobile phase for the illusion of the solutes. Where the isocratic illusion is performed by using a single solvent or mixture of two different liquids of same composition. It is called as isocratic illusion. And gradient illusion is nothing but in a mixture, there may be many solutes. There may be many solutes. Individual solutes may be eluted with different mobile phases. Say there are 10 solutes are present in a sample. All those 10 solutes are not going to be separated by using a single mobile phase. So what we can do that you separate two solutes by using a single mobile phase, and you can separate other four solutes by means of the second mobile phase. and you can separate some of the solutes which are present, which are left over unseparated by you by means of other mobile phase. So you will be varying the composition of the mobile phase from one mobile phase to another mobile phase, from one group of solvents to other group of solvent, that is called as gradient illusion. Apart from gradient illusion, HPLC operation will be done in two ways, and this is with respect to the stationary phase. One will be a reverse phase operation and the other one will be normal phase operation. This definition will be given based on the polarity of the stationary phase and the mobile phase. First, let me take normal phase HPLC, normal phase high performance liquid chromatography. This normal phase high performance liquid chromatography will utilize normal phase packing material. Packing material means it is a stationary phase. What do you mean by this normal phase? Normal phase means it is just like a silica material. The property of the stationary phase is comparable with silica, or silica itself going to act like a stationary phase. Means, if you look at the property of the stationary phase, the stationary phase is going to be polar while performing normal phase HPLC separation. The stationary phase is going to be polar. The stationary phase is going to be polar while performing normal phase operation. Say for example, water or triethylene glycol supported on silica or alumina particle will make the stationary phase. Apart from silica I am speaking, because liquid chromatography require liquid stationary phase. The liquid which is present in the interstices of the solid is water or triethylene glycol. Water or triethylene glycol is going to be immobilized into the interstices of either silica or alumina. Alumina is the supporting medium, so our silica is also a supporting medium where water or triethylene glycol will be acting like a stationary phase. Because these two substances are liquids, therefore this stationary phase is called as liquid stationary phase. So polar stationary phase, water and triethylene glycol are highly polar and they are immobilized over the surface of silica. Therefore, this stationary phase is called as polar stationary phase. And the mobile phase, what you are going to use during the elution step is going to be non-polar. It's going to be non-polar. Say for example, hexane, isopropyl ether, N-hexane, all these species are non-polar species. They are going to be used in order to serve as the mobile phase by utilizing their non-polarity. They are not polar, they are non-polar. This type of chromatographic separation is called as normal phase chromatographic separation. Simply we can denote this as NPHPLC. So NPHPLC or normal phase liquid chromatography is nothing but where the separation of the solute molecules are going to be performed by using a polar stationary phase and non-polar mobile phase. As an example, for polar stationary phase, triethylene glycol or water immobilized over the surface of silica or alumina, and non-polar mobile phases may be hexane or isopropyl ether or N-hexane, whatever it is. So here, while performing the normal phase separation, normal phase HPLC, whichever the solute which is going to be non-polar, or least polar, which will be eluted first, because the stationary phase is going to be polar and all those polar solutes are going to interact with the stationary phase for longer time. Therefore, their average residence time or retention time is going to be larger when compared to intermediately polar and non-polar solutes. So mobile phase is non-polar one, and if there are any non-polar solutes are present in the sample during the elution step, mobile phase will carry all those non-polar solutes out of the chromatographic column. Therefore, non-polar solutes will be eluting first, then intermediately polar solutes, then at last more polar solutes will be eluted while using this normal phase chromatography. In reverse phase chromatography, it is very simple. It is very simple. As I showed during the presentation of the structure of the stationary phase, the R group, in disillyl ether stationary phase, there was a substituent R. if that R was octyl or octadecyl group, you can look at here octyl C8 or octadecyl group, R is replaced by octyl or octadecyl group, the stationary phase will become completely nonpolar. So in normal phase LC, liquid chromatography, stationary phase was polar. In reverse phase HPLC or reverse phase liquid chromatography, the stationary phase is going to be polar. We are going to reverse the polarity of the stationary and mobile phases. So in reverse phase chromatography, the non-polar stationary phase is going to be used. Particularly, a hydrocarbon introduced silica can also be used as a stationary phase, and a relatively polar mobile phase such as water, methanol, acetonitrile, or mixture of any two or mixture of any three solvents can also be used as a mobile phase. This is going to be polar. So in NPHPLC, stationary phase will be polar, mobile phase will be non-polar, where the solute molecules are dependent upon the increased polarity they are eluted. Means first less polar or non-polar solutes are eluted, then at last polar solutes are eluted in NHPLC. In RPHPLC, because the stationary phase is going to be the non-polar, therefore non-polar solutes are going to be eluted at last. So if you look at the articles or the textbooks which are published on chromatographic techniques, most of the separations are performed by using RPHPLC, because RPHPLC is the most suitable one in order to elute the solutes in an array dependent upon the polarity of them. Polarity of the solutes will be varying. It is highly impossible to expect two different solutes are carrying a same polarity. So it is uncommon to say that, untrivial to say that. So if you go to pharma industry, 90% of the pharmaceutical industry will be utilizing this RPHPLC principle. So most RP separations are going to utilize the polar mobile phase, and these polar mobile phases will be adjusted to a required pH. And what should be the pH then? The pH which is going to be maintained must be less than 7.5. Must be less than 7.5. What will happen if the pH of the mobile phase is greater than 7.5? Because we know the property of silica, modified silica we are going to use as a non-polar stationary phase. And this silica tends to undergo dissolution if you increase the pH above 8. So in order to avoid this unwanted dissolution of silica in the mobile phase, we have to maintain the pH of the mobile phase at least less than 7.5. We should not go beyond 7.5 pH while using reverse phase stationary phase, such as octyl silane or octadecyl silane stationary phase, because these silica particles are going to be degraded if you maintain pH of greater than 8 or greater than 7.5. So, in contrast to the normal phase HPLC, as I told you, the most polar components appear first, and increasing the mobile phase polarity increases the elution time, because stationary phase is going to be non-polar, and if you increase the polarity of the mobile phase, the elution time is also going to be increased. This is a common sense we can understand. So you can see here the set of chromatograms presented for the separation of three species A, B, C by using normal phase chromatography and reverse phase chromatography, and also by varying the polarity of the mobile phase. By varying the polarity of the mobile phase. So, if you look at the order of elution while using normal phase chromatography, means the stationary phase is polar and the mobile phase is non-polar. If you look at the chromatogram, out of species A, B, and C, species C is eluted first, and B is eluted, then at last A is eluted. So the retention time of A is greater than B, it is greater than C. So polar stationary phase and non-polar mobile phase, if you are used, CBA is the order of elution. If you change the polarity of the stationary phase from polar to non-polar one, the non-polar stationary phase will become responsible to reverse the order of elution. Before, while using normal phase stationary phase, the solute C, which was non-polar, that's what it was eluted first. In reverse phase chromatography, the stationary phase is non-polar and hence A is more polar. You can see the polarity of solute A in this particular chromatogram. A was more polar, that's what it was eluted at last. Here, A was more polar, stationary phase is non-polar. There is no greater interaction between the non-polar stationary phase and more polar solute. Therefore, more polar solute A is going to be eluted rapidly. B will maintain its positioning when compared to normal phase separation because it is going to be intermediately polar species, and C is going to be the last eluted species because this is going to be the most non-polar species, and the elution time is greater for solute C because the average residence time of solute C or the interaction of the solute C with the stationary phase was expected more. Okay, and even medium polarity mobile phase if you use in order to perform NP-HPLC and RP-HPLC, you can see the overlapping or the degree of separation of the solute CBE. Medium polar mobile phase if you use, there is no baseline resolution is obtained here. There is no baseline resolution. And the same mobile phase is also used here also, and the baseline resolution is not satisfactory here also. The order of elution remained intact, but even though the order of elution remained intact, but the degree of separation or the degree of resolution is not acceptable. So while using normal phase packings means polar stationary phase, use non-polar mobile phase. While using non-polar stationary phase, use more polar mobile phase so that you can achieve the required separation with greater efficiency. So apart from this, separation of the solute once completed, it is very essential to find out the information about the solute, what kind of the solute which has been eluted out from the chromatography column, or what is the quantity of the solute eluted. So in order to get these two informations, detectors are required. So unlike as we have discussed under gas chromatography, detectors are also very useful in chromatographic techniques for either identification of the solute or quantification of the solute. Both the roles are played primarily by the detectors. Of course, column is going to be the heart of the chromatograph. After the separation is over, the quantity of the solute or type of the solute present in the sample will be derived from the signal given by the detector. So unlike gas chromatography, liquid chromatographic technique doesn't have any universal detector because this HPLC will be used for the separation of all variety of the compounds. It may be a simple molecule, it may be a macromolecule, it may be ionic molecule, it may be inorganic nature, it may be plant origin, it may be biological origin, it may be a clinical sample, it may be an environmental sample, whatever it is. All those variety of the samples can be subjected to separation of the solutes by HPLC. So in GC, only the species which are able to be converted into gaseous form, they can be subjected into separation by GLC or GSC. So we had universal detector, that was thermal conductivity detector, and dependent upon the principle of the solutes which they behave during the separation process, the detector will be functioning. So likewise, in liquid chromatography, we don't have such universal detector because the solute property is going to be varied from one solute to another solute. So therefore, this area is going to be the major challenge in developing the universal detector, unlike in gas chromatography detectors. So, dependent upon the response of the detectors for HPLC are classified into two types: bulk property detectors and solute property detectors. Difference is very simple. In bulk property detectors, the detector is going to give the signal with respect to the mobile phase bulk property. It may be refractive index or dielectric constant or density or electrical conductivity or thermal conductivity or fluorescent intensity, whatever it is, or absorption, UV visible absorption, whatever it is, the bulk property of the detector it is in respect to the mobile phase property. The refractive index or density or dielectric constant or viscosity of the mobile phase is differentiated from these properties of the solute molecule. Then the differential value between the refractive index or dielectric constant or density of the mobile phase and the solute molecule will be obtained in order to identify the solute or in order to quantify the solute. This is whenever we use bulk property detectors. In case of solute property detectors, as the name is implying, the detectors are going to function in such a way that the property of the solutes, such as UV absorption capacity, fluorescent ability, diffusion current, which may be obtained after undergoing oxidation or reduction of the solute. So these properties are not going to be exhibited by the mobile phase. Mobile phase do not exhibit any absorption in the UV region or visible region while using UV absorption detector, or mobile phase do not exhibit any fluorescent intensity. The mobile phase do not yield any diffusion current. So all these parameters, all these resulted values are only due to the solute participation in the stream. So as the solute reaches the detector, if it is a UV absorption detector, it will show the absorption, what is the amount of the light energy absorbed by the species. And this may be due to the conjugation. The length of the aliphatic chain or the number of aromatic rings present in that particular compound. So are the number of conjugated bonds present in that particular organic compound, whatever it is, D electrons, F electrons present in that. So they decide the color, or they decide the absorption in the UV region or in the visible region. They have the capacity to absorb the light radiation either in the UV region or in the visible region. You know Beer-Lambert's law for this. I need not to explain further about this. So those solute property will be measured by means of UV absorption detectors. Fluorescent detectors are most suitable to measure the fluorescent intensity once the compound undergo fluorescent excitation to emit light radiation. What is the intensity of the light radiation? That is going to be the characteristic one. And similarly, the diffusion current is also going to be measured. And this is as a result of the analyte diffusion because of the reduction or oxidation process. So, of course, all these things are collectively measured by means of typical detectors such as UV absorption or fluorescence or diffusion current detectors. Therefore, these detectors are called as solute property detectors. So, do remember, for HPLC, detectors are classified into two principal types: bulk property detector and solute property detector. Bulk property detector is functioning based on the property of the mobile phase or the bulk property of the mobile phase, such as refractive index, dielectric constant, or density. In contrast, the solute property detectors responds to the property of the solutes such as UV absorption, fluorescence ability, or diffusion current that is not possessed by the mobile phase.